Well, greetings everyone to the Little Lunch on the Prairie webinar series that was created for local resource managers by input they provided regarding our efforts to protect the prairie through the Minnesota Prairie Plan. And these webinars are being recorded, so if you'd like to ask a question, print it into the chat box, and at the end of the presentation I'll be able to read off the questions for our presenters. We don't show all the participants because this apparently improves our connectivity, but rest assured you're not the only one tuning in. We have a good group today. And I'm pleased to have Ian Lane, U of M PhD student, with us today. He received his undergraduate degree in crop and soil sciences at Michigan State University, but was fully converted to restoration during his time working on a study that investigated different oak savanna restoration techniques and their effects on pollinators. So we're excited to hear about his work with bee communities and restorations and remnants. And thank you so much, Ian. I'll turn it over to you.
Great, thanks a lot. So today I put together this presentation for you guys and girls to kind of go over this project that I've been working on for a couple of years now. I'm going to start off talking a little bit just about the science of community ecology, which may or may not be reviewed for some people, but I find it's kind of useful to kind of define what I'm talking about. Then I kind of proceed to talk about how I'm applying it to our restorations here in Minnesota, and I provide a little bit of our preliminary data that we've got from this past field season. So without further ado, so you guys have probably heard a lot about bees, probably not just from people like me, but from the media and all over the place. And I always love to just to rehammer this idea over and over again to everyone, is that bees are just incredibly diverse. You know, we talk about maybe like the rusty patch bumblebee, this endangered bee that was recently listed, or honeybees, or these single species. But in reality, we have hundreds of species of bees in Minnesota. In the entire world, there's probably 20,000 species, about 4,000 for the U.S. And in Minnesota, we don't know yet, but we're probably pretty close to 450. Which is some important work Crystal Boyd has been working on, trying to get us a full species list. But it's important to know that these guys are very diverse and they can be very different from each other. And so that makes kind of figuring out their conservation pretty tough. You know, do they need conservation even? Is like a really tough question to ask. Some species may, some species might not. So this is kind of where I find myself when I'm thinking about conservation and what we can do to help bees. So when I say community ecology of bees or bee communities, I'm usually thinking about like the suite of species that you might find in a particular spot. So each of these red blotches in this landscape represent like a prairie or some sort of habitat that's pretty good for bees. And in each of these little patches, we may find different kinds of bees. You know, maybe in one spot we find, you know, kind of an even number of species of different species, but maybe in another we find a lot of just one. And so if we think about all these patches together, we have some idea of how many species they are, but if we only looked at one at a time, we'd see very different patterns or might draw very different conclusions about which species are rare or are associated with certain habitats. So that's what community ecology is kind of concerned with. How do we understand the patterns of species that present themselves at different sites? And how do those sites affect which species show up too? Kind of like the converse of that question. So So kind of like using this idea, like how do we understand patterns of bees at different locations? There's a couple different questions we can start to ask. One that's really important for this is, what species are rare or common in different places? The picture here on the left, you see maybe a prairie. And there's a grouping of species. You have like green bee here, yellow bees here, and blue bee here. And if I were to go out and find all these, I might think, wow, blue bee is kind of rare. It's not common, but yellow bee is really common. But in the forest, if we go over here, we'll find that blue bee is actually quite common, and yellow bee is not common. So yellow bee or blue bee are not rare per se, but they're just different habitats are have different quality for them. So this is part of trying to understand how these different patches can hold different species. And the real question here is, what's green bee doing? Like, is he rare or she rare? Is it something that only occurs in one habitat? So this is one type of question we use community ecology to ask. Others are, what sort of factors explain the type of diversity we see at a site? So one great example that might be familiar to a lot of people is, how do different management practices change the species and the quantity of that specific species that we see? So hypothetically, I'm not saying this is really the way it works, grazing and burning, so don't walk away with this idea that one is better than the other from this, but let's pretend that if you graze a site, you end up seeing this collection of species on the left, which looks pretty good. There's at least 3 species, and 2 of them have equal representation. But on the right, you have this burn patch, and after you're done, you see, wow, yellow species really did great from that, but the other ones disappeared. So understanding how these different factors change diversity is another thing that I think a lot about with community ecology. So one of the things that I'm really interested in is applying these ideas to our restoration techniques. So kind of going back to that first question, how do communities and restorations compare to other habitats? So if we put in a prairie restoration, we spend a lot of time, trying to make it diverse and have forbs in it. Do we see communities of bees in there that are similar to other types of habitats, such as like remnant prairies, or are they their own community? Do they have their own kinds of bees that are unique to them, or maybe represented in different quantities in them? And that can give us an idea of maybe some bees might be really common in there, but rare in other sites. And it will kind of give us a sense of how good restorations might be for certain species, but maybe they're not so great for other species. And the second is, can we change communities to better reflect our goals? So higher diversity, more rare species, these are some of the goals we might have for a site that would be pretty common for restoration and conservation biology. And that's kind of like, does grazing increase the diversity of bees at a site or something like that? Those types of questions. And a quick anecdote, the picture that you're seeing is my field manager from this past season. She looks like she's having a great time, but she is not. This is a site that is really tall and just beautiful, but she has to swim through it like it's water. So it's just one of those examples of a restoration that went really well, maybe too well. So kind of sitting down and thinking about these 2 questions, I have to start thinking, well, what things affect the types of bees we see at a site? And how can we ask a question about one of those things? So this is kind of, I just invented this, I made it up, thinking about all the things that could potentially affect which species and how many show up at a site. So plant community is probably one of the biggest ones that we think about. How do the different species of forbs and other types of plants that we install affect the bees that show up? Planting maturity affects plant community, but also might affect just the bee community composition. Maybe as the site gets older, it gets more overgrown, it's harder for them to nest or something to that effect. Site management, like I've kind of said before, might affect the plant community. There might also be an arrow here, it might directly affect it, like maybe fire burns some of the bees in their nesting sites. And that's why, nesting resources is this other factor that's really important to bee biology. Spatial configuration and landscape are kind of allied in this different direction, in that we're thinking more about how does the placement and orientation of a site within the broader landscape affect bee community composition. And so that's kind of where I've honed in as being a place where we don't have great information about this question specifically. We have a kind of a vague notion that agriculture is probably bad. If you have a ton of agriculture surrounding your site, and it's like you have one restoration kind of alone in a big city, that's not going to be good for gathering species that you hope to conserve. But we don't actually know that for bees. We only have that vague notion. So I decided that this is something that could really benefit from some studies, so we can understand if we should be using certain kinds of placements, or if it doesn't matter at all. These are things I think that are really helpful to know about this. So kind of taking this question, how does the landscape surrounding a restoration affect the bee community composition? So to kind of think about this, I usually think about landscapes. I break them into two kind of really big groups. On the left, you have kind of the human-altered part of the landscape, which could be agriculture or mining, which is really big in certain parts of the state. And there's the natural part of the landscape, which doesn't necessarily mean good. So you have, natural habitat up here, or pastures or things. You might also just have cattail marshes, which are great for some things, but maybe not great for bees. So that if I were thinking about what elements of the landscape help bees, I might think that, well, a big wetland may or may not be great, but a grassland for sure is probably really good. So this is part of the uncertainty of what do these different parts of the landscape do to affect the bee community composition. So I set out to ask two questions for this past summer and in future summers. And to kind of lay them out really succinctly, the question one is essentially, how does purely agricultural, just focusing just on this one type of landscape cover, how does the amount of agriculture surrounding a site affect the bee community within a restoration? So I kind of depicted this right here in this picture. This kind of circle around this restoration here represents an area of measurement. So I just quantified how much agriculture is in here, and we're going to ask some questions about this particular feature. The second is, does increasing agricultural land use around restorations affect how similar remnant prairies and restorations are to one another in terms of their community composition? So that might affect, a community of bees in a restoration may be affected by remnant bees. They might be trying to access the site and disperse out of it. And if there's high amounts of agricultural land around the site, that might affect their ability to reach it. And therefore, the community of bees as a restoration may not be quite the same as in a remnant. So these are kind of two related questions. One of them is just, how do restorations differ as their amount of agricultural land changes? And the second is, how does a restoration community compare to a remnant as the amount of agricultural land changes? So to ask this question, I worked really heavily with the Board of Water and Soil Resources, and I'm not sure if John Boz is in the audience, but I have to give him a really special shout out for his help in contacting landowners, getting site information, and coordinating with the other people who help manage these easements. He was critical, and so there may be others of you that have helped provide information, and I just want to let everyone know I really appreciate it. It's already becoming really useful, and I can talk about that later maybe, but for other people besides myself. But using this information, I was able to come up with 70 potential sites. I selected around 3 core areas, or areas that I've kind of referred to as the Blue Stem, Ordway, Blue Stem area, Ordway area, and the Chippewa area. And I wanted to kind of make sure that my sites were nested within these regions because I wanted to make sure that there was actually remnant prairies from which potential colonization could happen. I didn't want to just throw it into the middle of an area that had no remnants, because that might just create a really unusual kind of community. So what I did, kind of going back to that first picture, was I drew a circle around every site, all 70 of those sites. It's about 1.5 kilometers from a radius from the site border, and in that radius, and so I'll also say that the reason that I picked 100 or 1,500 meters or 1.5 kilometers is that a lot of the previous data shows that bees really respond to landscapes within that range. So anything more than that doesn't really help me understand what might be going on. And it might be related to dispersal. People don't really know why this special number is the one that seems to be most predictive, but that's the basis for why I use this number though. So I essentially drew this area of measurement, and then I calculated how much agricultural land is within the buffer from this site. So here you can see blue is non-agricultural land and olive color is agricultural land. And then I calculated how much of this area of measurement is proportionally covered by agriculture. So it turns out this site has about 77% of its area around it into agriculture. So I eventually, through a long process of trying to control for different factors within these sites, like how old is the site, how big is the site, things like that, because those are all things I kind of tried to make sure, because they could affect the community composition. And I wanted to just focus on this one question, landscape, how does that affect? So I didn't want other things interfering with that. So I actually got about 16 sites, kind of from all these different regions across this area, and not pictured here are 5 remnants that I also picked. So I chose this area up near Blue Stem to kind of focus the as a focus for the second question. How does the surrounding amount of agriculture affect the bee composition compared to a remnant? So I have 5 restorations here, and each one is paired with a remnant in the landscape that's nearby. And so you see up here we have a site that's almost 90% agriculture, and some that are as low as 25%, up to 40, 50, 60s, almost 70s, and then 90s. And so the reason that we paired these sites with a remnant, this is something that's actually, this is great that we were able to do this. And I think Minnesota is really special in that we have enough remnant sites that we can actually do a paired design like this. It's something that not a lot of people can do. It allows us to kind of control for a lot of contextual things. So, you know, if I were to sample just a bunch of remnants from all over the place, they could have different bee communities due to different types of properties of the environment. Like maybe the soil texture is radically different, you know, or something like that from the restorations. And so this really is great that we're able to kind of compare them to sites that are really only about 5 kilometers away. that's what I was shooting for, 5 to 7 kilometers is the distance. And so they're not necessarily independent from one another. And so hypothetically, bees could be colonizing these sites from these remnants. So this past summer was our first kind of big data season, and I'm going to kind of try to describe how we set about to measuring the bee community within these plots. So within each restoration and remnant, we designated a 1 hectare plot. And this hectare plot was the focus of our sampling for the rest of the season. So we used a couple different methods sampling. We used passive samplings, and we tried to do it as frequently as we could. We had about 21 sites, and we were able to do about 3 weeks. Every 3 weeks, we were able to visit every site. So about 6 samples a season, we ended up getting about 5 samples, actually. But you can see here there's these passive sampling, these bee bowls, which is something you might have heard of before. They're filled with soapy water, and bees find their way in like they're artificial flowers. They get trapped and drowned and die. We also use blue vein traps, which they have a similar idea, but they collect different kinds of species. So I'm trying to find as many different ways of capturing species that have different biases so that I can get the best picture of what the community looks like. And so the last way that I was doing sampling for bees is through hand netting. So essentially, this is an active effort where we walk transects down from one end of the hectare to the other, so about 100-meter transects. And we put an hour of effort in the morning and an hour of effort in the afternoon. And we essentially just walked down these transects and collected bees when we observed them visiting a flower. And then we recorded which flower we collected it off of and which transect. And so this type of information doesn't only give us information about the bees, but also the flowers that they're visiting. So that's really great. We can get some flower association data, and we can start to understand how the floral community might be influencing these sites, as well as just the landscape. So we also collected plant community data from these sites because we think it could be really important. We did this along the same transects that we netted down, and we did it with 1-meter quadrats. So we'd place a quadrat every 10 meters, and we'd count the number of blooms of each species within there. So we looked at kind of abundance and richness within each quadrat. And so we had 6 transects total, and so that equals about 60 meter quadrats. We got a good amount of plant data from these two. So to kind of give you a quick summary of the field season, we collected about 11,600 bees, which is pretty good for a study like this. And we hand netted about 7,200 of those from 90 different plant species. So there's a lot of plant data and plant interaction data that we got, which is really great. To take a quick peek at that, these are different species of plants in these restorations, and I cut out all of them except kind of the top. And so what you're seeing here is how many individual species we caught from bees, individual bees we caught from each species of flower. So this is super surprising to me. I had no idea that golden Alexanders here on the left was going to be like 1,800 individuals caught off it. It was crazy. I never expected golden Alexanders to be that important. And then we see some other species, the Retibita pinnata, the gray-headed coneflower, and purple prairie clover were very attractive. And then this was a surprise. The Ridgeron philadelphicus, one of the perennial fleabanes, was really attractive too. And it bloomed at the same time as Zizia. So, and then some of these other ones are pretty interesting, like Cirsium is a weed. It's Canada thistle, and yeah. So there's we have about 120 plants in the system, and these are kind of like the most important players, it looks like, for the whole study so far. Kind of looking at restoration and remnant bee catches, the remnants really didn't have a lot of bees compared to the restorations, but and that's kind of depicted here on the left. Remnants, we only caught about 600 bees from them, but from the restorations, we caught, and these are only comparing the paired restorations, so 5 sites versus 5 sites. We caught almost 2,000 from the restorations here. So this is a crazy difference between these two habitat types. But the remnants, we collected far more bees off of more types of flowers. So there was just more species of flowers there, and the bees kind of came from a broader selection of them. So that's really interesting too. And so this is something I'm going to try to dig into as the year kind of goes on. So to kind of get at that species level information, there's a lot of work to be done, and this is something I hope to come back and share as we get more information. This is us sorting the specimens that we collected from this past season, and so the identification process is really tough and it takes a long time, but we're already getting some pretty cool data, especially on bumblebees, which is like probably one of the easiest to identify. So with that, I think I'm out of time. So I don't know if people have questions. I don't know how this works with the webinar.
Get myself unmuted here. Yes, I encourage people to type in questions. I had one there, and see if I can articulate it. So the last slide, when you were talking about the number of bees collected, yeah, the remnant, it's the last chart there. A little more clarification on that for me would be good. So
sure.
You had more plants and in the remnants,
okay.
so therefore you collect them.
Yeah, so yeah, I kind of blew by this. I was kind of worried about timing. So I'll try to describe this better. So there's no means or any type of calculation. It's simply I summed the total number of individuals that we caught from remnants and the total number of individuals I caught from restorations of only that subregion near Bluestem Prairie. So this is kind of just a way of saying the total number of bees and remnants was far lower than the total number in restorations. That may differ a little bit when we start thinking about, okay, what does time of year do? What are these different, so maybe there was no bees in the remnants in certain times of years, but they were really present in other times. That might look a little different when we compare them to restorations. The one on the right is a little bit more complicated in terms of, so when we were thinking about, when we are catching bees off of plants, we noted the species of plant it came from. And so when in remnants, we found far more bees on more types of plants. So we found about 70 different plants that bees were using in remnants, compared to only about 40 or so plants that bees used in restorations. And that could be because there's just more species in remnants than there are in restorations. But I'm not sure if that's really true. The restorations did have a lot of species when you kind of thought about the planted species plus the weeds. They may have had about equivalent numbers of species if you considered weeds and the intended plants together. But they tended to be on more types of plants in the remnant. So I thought that, so this is almost, the plot looks a little weird. It's almost a third more plants were being used by bees in remnants than restorations. So I thought that was like a really interesting observation from this data that we have. Does that help? Did that kind of clarify it?
Yes, it did. And I assume the diversity of bee species is something you're getting information on now as you identify the...
Yeah, that's really the star of the show. I'm not so much interested in the number of individuals per se, but more which types of individuals tend to be more common in different kinds of habitats. So once we get that species information, that's really going to be key for kind of figuring out what's going on. So we may have caught fewer bees at remnants, but there may be more species or different kinds of species than in the restoration. So I'm really excited to get that data back and to come back and share it when I can.
Yes, definitely. Fred Harris asked if you were, if you know how the remnants were being managed or not managed.
I have some of that information, so I can So the remnants that I used, 2 of them were owned by the U.S. Fish and Wildlife Service, and I was in contact with Becky Esser about those to a large degree, and she has great information about the history, and we communicated a lot about what their management actions they were going to take and things like that. So we had one site that was a WMA, a wildlife management area, which was Clay County WMA, and I don't have as good information about, it may exist, but I'm not 100% sure what their management was yet. The area at Bluestem is quite well documented, and I think that it hasn't been burned in quite a while. In fact, I don't think most of the sites have been burned recently, within the last couple years, maybe even 3 years. None of them are grazed either. I'm not sure if any specific weed management actions are being taken on those sites other than those kind of broad management, but yeah, that's all information that should be pretty available for this.
Great. Well, we've got quite a few questions, so we'll keep going down the line. Lisa's asking, are you recording soil types?
No, I haven't been. Though that's something that, 2016, I spent a lot of time scouting sites. And I think the thing that most kind of blew my mind, I think, is that a lot of these easements are just really a large upland component, and then there's like a pond with a kind of a wetland almost community down below where there's lots of willow and things like that. And so I tried to always place my sampling hectare in the upland portion, which isn't saying I controlled for soil type at all. But I was definitely trying to shoot for that upland community, trying to stay away from kind of that more, not that I think it's better, but just for consistency, so I can compare between similar communities. And so that's kind of how I try to accommodate these different moisture classes and things. But yeah, I do know that some sites are really sandy and that can really affect the plant communities, and that could also affect nesting. So it's something that I've been thinking about for sure. But I wasn't able to kind of filter my sites based on it.
Well, the good news is soil type's always available, so it doesn't change much. Yeah. Chris is asking, any idea as to the variety of bee species collected? It's kind of the same question I had asked from remnant versus restoration, and we will be sure and keep in touch with you and have a follow-up when you get a better idea of that.
Yeah, I can say some real general things if you'd like.
Sure.
So we do have a lot of the bumblebee data already, and I can't necessarily say if there were certain kinds of bumblebees that were more common in remnants versus restorations, but I will kind of say generally that I was totally blown away by the number of species that are considered rare that we found. So one of them, Bombus pennsylvanicus, is kind of nationally considered to be declining. We found a good number of them. Bombus fervidus is another one that is reported as being rare in Ontario and other parts of eastern U.S. And we found that's like one of the stars of the show. Bombus borealis is something I've never seen before, but we collected it in great quantities, and it's totally fascinating that it was so abundant in some of these sites out there and you just never see it in the Twin Cities or over in the eastern part of the state. So there's definitely some characteristic species that are really pretty prairie oriented and people think are declining, but maybe they're not declining in our areas as much as they are in others. So I think that's good news. That's positive. I didn't find any rusty patch, so nobody from the U.S. Fish and Wildlife Service needs to worry. But so I'm not sure what the story with that is. But yeah, the species information we caught some really interesting things, and I'd also say that there's some species that seem just ubiquitous. Like there's some species of sweat bee, they're everywhere. They're in the restorations, they're in the remnants. Nothing can stop them. They're definitely doing great. They're doing really well. So yeah, so those are some quick generalizations. But yeah, I'm really excited to kind of get more specifics as we get through that identification process.
Yeah, it looked like you had quite a few to get through.
Yeah, 11,000. Yeah.
Phil is asking, were there any honeybee hives near your site?
Some of the restorations, yeah, that was not uncommon. I wouldn't say it was like ubiquitous or anything, but some people did have honeybee colonies near the restorations. And we did try to keep track of honeybee visits to the sites. And we do have that information. I will say that it's kind of like probably not the best of all the data we collected. We tried to keep track of it, but at the end of the day, I wouldn't try to publish any of that information. But I was really surprised how much honeybees use bee balm, the Monarda fistulosa. I was under the impression that the flower was too long for them to use, but we found lots of them on it, as well as the Anis hyssop, the Augustaceae funiculum. So they used the sites to some degree for sure, as well as the Cirsium. We found a lot of them on the Canada thistle.
Well, I'm not sure we'll be adding that to our restorations anytime soon.
But if you could stop it, right?
Yeah, right. Final question, and Krista, she did come in late, so you perhaps answered this. Did you determine relative abundance of plant species in the survey areas to calculate relative important values for each plant species?
Yeah, so we actually did two things. I didn't actually say this in the little presentation. We had those transects. We had 6 transects set up within our hectare area, and we did 10 quadrats per transect. And within each quadrat, we counted the number of flowering units. So that changes a little bit based on the type of species you're thinking of. Some of them are really obvious, some of them are not so obvious. And so we did get relative abundance of flowers in that way. We didn't keep track of anything like grasses, which are also important. I don't want to take away any glory from the grass, but we also tried to do just species occurrence. So we also, down our transect, we recorded every single species that we saw, not whether it was in a quadrat or whether it wasn't. And we did this for each transect anew. So what I mean is, if we saw, let's pretend that I saw some kind of hawkweed down one transect, I would record that. If I saw it down transect 2, I'd note that again. So we have kind of a rough measurement of frequency, even without the abundance, for when we did. So we have relative abundance from our quadrats, and we also have like a rough frequency from our pure species counts down each transect.
Wonderful. Any final questions? I think I've gotten down the list here. Very, very interesting presentation, and I think relevant to a lot of our work in the prairie areas, looking at this sort of agricultural landscape and restorations. So thank you so much, Ian.
Yeah, it was a pleasure.
And we'll look forward to having you back. Next week is the last of our Little Lunch on the Prairie webinars, and Mike Warland and Darren Carlson of the DNR will discuss their project, which is focused on long-term prairie monitoring. So thanks to everyone who joined today, and we look forward to having you join next week if you can. And again, thanks, Ian.
Thanks again.
Bye.
Bye.